This video is cell cycle cyclin CDK complexes. The cyclin CDK complexes are part of the cell cycle control machinery, and basically what they do is control the cell division machinery. And so basically each cyclin CDK complex will phosphorylate a different set of target proteins, and that triggers other machinery. So for example, one complex of cyclin CDK would trigger DNA replication machinery, and a different cyclin CDK would trigger something else. Basically what this means is this is going to coordinate the cell cycle control machinery with the cell division machinery. And overall, this cues up or triggers a transition in the cell cycle. So this one's a little scary. I put the scream up here. I hope Edward Munch doesn't mind me using that. Basically, we've got a big old graph here. This is what you got to memorize. So the cell has many different versions of cyclins and CDKs. This is a take-home message. And as a reminder, each of those versions of cyclins and CDKs works with a partner, and those partners stay together and facilitate a particular process, and they're not always bound to each other. They can split up, and as you know, cyclins will come and go. CDK stays the same, but the only one individual, like Robin, always works with Batman. Robin doesn't work with R2-D2, or C-3PO for that matter. Okay, so just to clarify, it's this whole thing that you've got to memorize. You don't need to memorize these graphs down here. And basically, to run you through what is going on with the graph, we have the xenopus and non-mammalian versions of the cyclin and CDK written over here. So this is the xenopus slash non-mammalian cyclin M, and then the corresponding M CDK is the xenopus slash non-mammalian partner. And xenopus is frogs. The checkpoint that each partnership works with is listed here in the middle, and then on the far right there are the mammalian names. I'm going to warn you that I really use these ones a bunch, but you should still know the xenopus non-mammalian ones since they show up. And actually these ones are a little more straightforward to learn because their names coincide with the checkpoints instead of kind of this hodgepodge of numbers and letters. And just to kind of talk through that, so M cyclin and M CDK, those are controlling the G2 to M transition, and in mammalian cells, the counterparts are cyclin B and CDK1. Likewise, for the S to G2 transition, S cyclin and S CDK work together in xenopus, whereas in mammals it's cyclin A and CDK2. For the G1 to S transition, which is also known as the R point, there's the G1 cyclin and G2 CDK. And in mammals, they instead are called cyclin D1 and CDK4/6. Finally, for the G1 to S transition, that is just after the R point, then we have G1/S cyclin paired up with the G1/S CDKs. And for mammals, it's cyclin E and CDK2. Again, you don't need to memorize the graphs that are down here. This one on the left is from figure 18-8. In your textbook. This is not in our textbook, at least not in this reading. But the visual is really important and is one of the take-home messages. So basically remember that the cyclins are going to cycle in terms of the amount that's present, and so they're going to vary widely in when they're present and when they're not. And so the S cyclin, which is shown by this blue line here, is basically at no levels at G1, then increases as we go at the end of G1 into the S phase, hence S cyclin, and then it is at high levels until the M phase, and then it drops off. On the other hand, M cyclin is at essentially 0 levels all the way through G1 and S, starts to build up through G2, and then it reaches its crescendo, its high expression level at the end of G2, and that continues through M, and then it drops off again. Check out on the lower part down here how these levels of cyclins correspond to the active partnership of the cyclin and CDK. So for S cyclin, if we check that out here, so the S cyclin is paired up with S CDK, and here we see that S cyclin builds up at high levels. So once it gets to high levels, then that will be when it's activating the S CDK at its highest levels, and so we see that right here. And you can see the little red lines indicating that. On the other hand, for the M cyclin, so M cyclin is paired up with M CDK, and while these have similar shapes, notice that they're different colors from the other cyclin CDK. And so M cyclin, when it's at high levels here, then that means that it is going to have a high likelihood to be bound to MCDK, and that means that the MCDK is at its highest level of activity when its corresponding cyclin is at its highest level of activity. Over here on the right, we're looking at the mammalian cyclins, and we're just looking at the cyclins in this picture. And the big deal here is to see that we've got one cyclin that's spiking here, and you can see that that's spiking in the M phase. And if you go and check out what that is, so that you would expect would be in the G2 to M transition. So cyclin B over here does correspond to the G2 to M transition. And so what I'm really trying to show you here, and what this graph is trying to show you, is that it's no coincidence that when the cyclin spikes, then that's when we have the transition time or checkpoint. This is labeled nuclear D1, but this is a cyclin as well, and you can see that that cyclin spikes in G1, and so that cyclin D1 is used within the G1 to S transition, which is also known as the R point over here. Cyclin E, then you probably would not be surprised to see that that follows just after the G1 to S transition. This is the G1S. There's two checkpoints here. This one is after the R point, so R point you can see labeled down here, and that one spikes. Then cyclin A, which we can see over here, corresponds to the S to G2 transition. Cyclin A has a little different pattern of how it peaks from the other cyclins, but still notice that it spikes within the S phase and is basically kind of wrapping up its high expression levels at the S to G2 point. And then we see this other spike here that corresponds again to cyclin B, and so basically down here we see the names for each of the parts of the cell cycle, and basically we're repeating now, we're going from G2 to M. And if I haven't said this already, so basically to read these graphs, follow from left to right along here, and these are the symbols for the names of the phases of the cell cycle. So M for mitosis, G1, S, G2, etc. And over here, same thing, we've got G1, S, G2, M, and then we repeat back to G1. All right, and so I want to hit this very important take-home message and highlight it. So again, the deal is that the cyclins come and go in terms of their concentration. The cyclin concentration will determine which point of the cell cycle that the cell is in, not the other way around. So the cell does not determine the cyclin concentration. The cyclin concentration actually determines where it's at in the cell cycle. Each concentration of the CDK protein itself is relatively constant. However, the activity of the CDK is determined by the cyclin concentration. There's some pretty awesome experimental evidence for this, and if you want to check that out in your textbook, read pages 609 to 610.